All right. Hello, guys. All right, this video goes with your stem cell notes. So please make sure that you have your cell division notes document open and scroll all the way down to a page called stem cells. So stem cells are something that some of you may have heard about and some of you may have not. Let's kind of put in perspective how many cells are in our body. So our body has that many cells, which is a lot. That's 13 zeros, right? And those cells had to have come from something, some original cell. And the process of making all of these cells is actually not as easy as it sounds. So let's go back to the very beginning of you. So we all started as one itty bitty cute little cell. Your first cell that was ever you was the fertilized egg, right? So, which becomes fertilized typically a day or two after your parents had sex. This cell went through the cell cycle to produce a small clump of cells. So that original cell started reproducing like really rapidly, actually, and creates a clump of cells. Eventually, after about 10 days or so, that clump of cells then begins to do what we call differentiate into different types of cells. So it's actually, it doesn't take that long for us to start having like specialized cells, even though we were just a clump of cells. So the human body contains about 220 different types of cells. Without listing all 220, let's keep in perspective that we could probably make a long list of different types of cells. So in our body, we have blood cells, bone cells, brain cells, nerve cells, muscle cells, skin cells, and so on, right? Every type of cell in our body has a specific task or job or function. Also keep in mind, though, that like every cell in your body has a complete set of DNA. So your liver cells, yes, they're liver cells. They look different, they function different than other cells, but they still have a complete set of your DNA. So liver cells are complete cells that just have been triggered to act like liver cells. The same exact set of DNA is going to be found in your red blood cells. Red blood cells look very different than liver cells because they've been triggered to be shaped and to function like a red blood cell. They still have every cell in your body except sperm and egg have a complete set of your DNA. Heads up, I think this is the first mistake all school year, but there is not a spot, I don't think, in your notes document for this. So if you want to pause, please make sure, I added it, but I don't think it edited like your document now. Please make sure that you get this in your notes for Pete's sake. So the definition of stem cells are unspecialized cells that have the potential to differentiate into any type of cell. Okay, so these are cells. Stem cells are what we call a cell that really hasn't been given a job yet. It hasn't been triggered to act like any specific type of cell. Okay, so again, make sure this definition is in your notes. The term differentiation is what we call the process in which a stem cell develops into a specific type of cell. So we take a stem cell, make it a red blood cell. We take a stem cell, make it a bone cell. Take a stem cell, make it a liver cell. There's 220 different options for the fate of every cell in our body. So stem cells can differentiate into any types of cells. And remember, there's 220 different types. We call them stem cells because they have the ability to branch off into so many different types of cells. Oftentimes what this looks like is stem cells come from a clump of cells in a petri dish like we see here, and then we can differentiate in the laboratory into those 220 different types of cells. Let's talk about sources of stem cells. So Stem cells can be embryonic, which means that they're obtained in the early embryonic phases. So remember I said within the first 10 days. So after an egg has been fertilized, those cells reproduce like crazy into this clump of cells. But again, about by day 10, they're going to start to differentiate. They're no longer considered stem cells then. So, and we can get embryonic stem cells in a petri dish if we just take sperm, egg, they become fertilized. We give a number of days to reproduce stem cells, and then we have these clumps of embryonic stem cells. Another place that we can get stem cells are in adults. So stem cells are also found in our adult bone marrow. The problem is it's actually really difficult to get adult bone marrow. It's not something like you can't just go donate blood in one evening after school. You know, they can't just tap into the bone marrow like you can tap into blood. So although we know that adults have stem cells in our bone marrow, it's actually not like a common use because it's just so challenging to access. And then the third one is man-made. So scientists have recently discovered ways of kind of creating stem cells in a lab. So they basically de-differentiate or reverse the signals that turn specialized cells back into the stem cells. So, and this is kind of like the latest. So, and scientists are really excited about the ability to now take a clump of cells that have jobs and actually reverse them back to stem cells. So if we can do that, if we can take a chunk of cells, reverse them to stem cells, and then say, we need nerve cells. We can trigger them to act like nerve cells, and voila, we have a clump of nerve cells that we can now use in some type of cell therapy, which we'll see in just a second. Obviously, I think scientists are absolutely amazing. You could get totally lost on Google just looking at the latest news in the last couple of years around stem cell research. It's absolutely fascinating. Okay, so how it works is typically stem cells are placed in a living medium, like a petri dish. We usually would call that, well, a couple different things, but yeah, so imagine that they're plopped in a location, typically a petri dish, where they can live and survive and thrive. And they go through mitosis. So we let them go through mitosis until there are like that many stem cells, like lots of stem cells, like a huge clump of stem cells. Scientists then use special cellular signals to trigger and direct differentiation, which will provide that many specific cell types. So whatever type we're looking for, let's say we need white blood cells, right? We can trigger this huge clump of cells now to all act like white blood cells. And then we can use that in some type of cell therapy. So obviously this is a huge deal. We do have the potential to save millions of lives, and not only saving lives, but there's countless examples of people that are still alive, but by using some type of stem cell therapy, they may be able to gain a specific ability or skill or something. So an example of that would be paralysis. So we can potentially treat someone that's still very much alive, but we can treat their paralysis and then they'll be able to walk again, right? So many health problems involve the loss of specific cells. So remember, I kind of talked a couple days ago in a lecture about heart attacks damaging muscle cells. Well, with stem cell therapy, we have the ability now to end up with a clump of healthy muscle cells that we could potentially get really good at replacing damaged muscle cells in our heart. Strokes cause brain damage. Those are nerve cells. So we now have the ability to end up with nerve cells. Sorry, there's a typo. Spinal cord injuries again cause paralysis, and we're working on replacing those damaged nerve cells. Sorry. Okay. Some more like specific examples of types of therapy that we're feeling hopeful about is type 1 diabetes. So this is someone that's born with the inability, typically the inability to produce insulin. So type 1 diabetics are missing one type of special cell, and stem cells could be used to treat this specific disease. Parkinson's disease causes nerve cells to just disappear, get broken down and disappear. We can now produce nerve cells, so we could potentially treat Parkinson's disease patients with new nerve cells. And then again, we're looking at paralysis. So we talk, or for example, someone that has been injured in a motorcycle accident. There's two two modes with this. So it's also important to think about what we call human disease modeling. So basically, scientists could take stem cells from a particular patient and study the disease in that lab. So if someone has a specific disease and we want, the cells that are affected by that disease, if we want to use different treatments, right, we now can extract and essentially kind of like use those cells for different tests. Or with drug therapy screening, scientists would be able to test drugs, right, in a specific laboratory setting using stem cells to see the outcome of them on actual human cells. So keep in mind, we do a lot of animal testing, medications, like toiletries, makeup, all those things. Almost all of them are tested on animals in a laboratory setting, and now we can just test them on human skin cells in a petri dish. Oh, here's a cool example. So this is actually still 5 years ago. You may have seen this, but basically scientists used stem cells to grow a human ear inside of a rat. So basically, the process was, so they had this rat and it was a live rat. They created a mold of a human ear, and the mold itself was produced with a specific material that that broke down over time, like quickly, like within a couple weeks, I think. The actual ear mold dissolved. What they did was they took the ear mold and basically introduced it with a whole bunch of stem cells and then put it into the live rat. And so those cells need, like everything our cells need to live, right? So they can't just live like plopped on my desk. They need to be supplied with, obviously, everything other, to stay alive. So these scientists inserted the oh my gosh, what was I calling it? The ear, basically the ear, laced with stem cells, and inserted it into the rat. And what they realized was that the cells thrived. The cells got all the nutrients, everything that they needed to survive. They ended up covering the entire ear. They were mostly cartilage cells. The ear material itself slowly dissolved after a couple weeks, and within a number of weeks' time, they actually were able to extract it from the rat. It had human cells. It was an ear made of human cells, mostly, again, human cartilage cells. So it had the structure, the flexibility of like a normal ear. Amazing. There's just lots of examples of using stem cells. Here's one talk that focuses on athletes. So basically making, I think it talks about like growing ligaments and tendons and stuff for athletes that have damaged their, ligaments or tendons or something like that. I think it's important to notice that this can be a controversial topic. Traditionally, stem cells were retrieved using egg cells, so human eggs. So there were a lot of, I'm going to say mostly religious groups all across the country, but also globally, that are against human stem cell research because it is, human eggs. So for that reason, stem cell research has actually been pretty slow in progression because up until recently, even, like these research agencies wouldn't get any, for example, like government funding because it was so controversial. But nowadays, now that we have this understanding of how to, take a clump of cells and kind of reverse the differentiation, it's kind of lost its controversial issues. Just some more examples of stem cells in the news. Like I said at the beginning of this lecture, most of you have probably heard about stem cell research. The last thing I'll mention, I think, is, which is kind of cool, is that we also know that when a baby is born, the umbilical cord, it's also filled with a small amount of blood that contains high numbers of stem cells. So as a mother, when you give birth, you have the ability to sign away your stem cells for research. And you also have the ability to save the stem cells from the umbilical cord. You pay like a monthly fee, and they're kind of frozen, and you can kind of have them frozen in time for future therapy for your children. So, when you have a baby, if you save the stem cells, that at any point in time, there's, their actual stem cells are available for stem cell therapy. It's becoming like more and more popular. I think when I had all 3 kids at Kettering Hospital, and I actually do think it was an option for me to have the stem cells frozen and saved, but it was expensive. It would have been really expensive, overall so I didn't do it, but I think it's really, really cool. Okay. All right. I think that is it.